Yeah, welcome everyone. So today it gives me immense pleasure to introduce Dr. Prashanth and Suravajhala. He's founder of India's largest bioinformatics society, that is Bioclues, which is working for mentor-mentee relationship since 2005. As a distinguished figure in system biology and bioinformatics, Sir is a principal scientist at Amrita School of Biotechnology, Kerala. He holds a Ph.D. in system biology from Aalborg University, Denmark, with 7 years of postdoctoral experience across 4 laboratories. He is also principal investigator in 4 and more projects, questing keywords of hypothetical proteins, vitamin K, long non-coding RNA, and prostate cancer. And with 300 plus peer-reviewed publications, he is a passionate scientist dwelling into functional genomics, protein informatics, and interactions. And beyond academia, he is a global explorer and has traveled across 70 plus countries and authored a book titled Your Passport to a Career in Bioinformatics. So, with this very brief introduction, I'll request you, sir, to please take the session in week 6 of Bioinformatics for School Children program.
Yeah, thank you so much, for this gracious introduction. Thank you so much, Raju. I'm thankful, to you for organizing this program very effectively. So friends, welcome all. In fact, you're all our heart of, I would say, bioinformatics program. That we have been carrying it over the last, I would say, 4 years, successfully. Although we started it informally in 2008, as I was telling you, in the slums of Pune, as a part of our Teach India program. So today, what I'll do is, please feel free to stop me at any given point of time. Okay, I'm at your disposal. So we'll be introduced with something what we call it as systems biology. So if you are wondering, what is systems biology, what is biology? Logy is study, bio is about life, and a system is nothing but it can be any living system. When I say living, it can be a kind of a thingy like system, like a computer, which you may also call it as a computer system, or you might even take our body as a human body system. This can also be taken as a system. So the biology of a system, which means the life processes, what goes through within this particular system, is what we call it as systems biology. And then you might ask me a question: why systems biology is very important? What makes systems biology essential? So let's talk about this figure. Can someone identify this particular figure? Anyone? If you can make it a try, what do you see in the figure?
Sir, it's a human skull.
Okay, you think it's a human skull? Okay. Okay. Anything else? Any other things that you see from this cartoon? Yeah. No, it's okay. No? I think you're giving it a try. The cartoon is not that great, but what you could see from this here is, in fact, there are 7 men somewhere here. One of this other man, is standing just behind this elephant. This is an elephant. Okay. And of course, there is a human skull, but this is a kind of a visionary thing. So there is a very famous story about 7 blind men and an elephant. Okay, you know that, right? I don't know if you have heard of this. So what happens? Usually the blind men cannot see anything. They come up seeing a kind of a moving object. Okay, one man touches the head of the elephant. He imagines that it could be something like a like a soft bed. Another man touches the trunk of an elephant. He thinks that it could be probably something, a big rope. And the other man touches the wiggle of the elephant tail. Okay, he imagines that it's just like a soft leather. Other one climbs upon the elephant. Okay, and he thinks that it's a big bed, and he thinks that he wants to sleep. Okay, so this is the beauty of any study that we are always trying to work on. So what happens is, if you're not able to see what you are trying to understand, okay, you come up with a kind of an imagination. That imagination is very, very important in the realm of biology. That imagination is very, very important, as a budding scientist or as a budding researcher, or for that matter, any scientist. Okay. So, if you really talk about bioinformatics, right from day one when Raju started and till various other speakers, bioinformatics is a wonderful tool. Okay, it stems from two important words. One is biology, the other one is information technology. Okay, I need not tell you these wonderful words during this sixth session. Okay, but it's basically a tool. But the science behind this bioinformatics is what we call it as computational biology. Okay, which means you drive or you make use of computer systems to read out what's stored within this biological parameters. Okay, that is what we call it as computational biology. Okay, and obviously, when you talk about bioinformatics, when you read about what is the information that is generated from this biology, okay, you get a lot of data, isn't it? You get a lot of data, be it a DNA data, be it a RNA data, or protein data, any kind of molecular data, you keep on getting it. Okay, that particular study which we entail from any organism is what we call it as bioinformatics. Okay, so bioinformatics is a tool, whereas computational biology, or for that matter, it's other native disciplines like systems biology, is is a discipline. Okay, whereas bioinformatics is only a word, it's only a keyword, which we use it as a tool. And why? What does bioinformatics entail? It is a tool where we can store, transfer, analyze, and display humongous data that is being generated from these organisms or from these biological entities that we are trying to store. Okay, and then you might want to ask a question. Okay, then why bioinformatics? Can't it be simply biology? Why do we need that informatics? Why do we need to derive that particular data from the biology, from that particular organismal source? This dates back to the very previous talks. Maybe you might have been introduced to what we call it as a central dogma of molecular biology by including Rajal, Pushpendra, and others. Okay, can someone tell me? Can tell me what are the three important biomolecules in an organism? Anyone? What makes life, as an important entity? What are those three molecules? Can someone tell me?
Is one of them proteins?
Yes, of course, proteins. Wonderful. Yeah, protein is a very important aspect of an organism to sustain. Okay, very good. And what are the other molecules? How does protein? How do you get the protein? There must be something, right? Protein, is obtained. You get even a sweat also has a protein. Okay, how do you know, okay, that this is a protein that you're getting? What is a predecessor for that particular protein? What is a prelude to that particular protein?
Amino acids?
Of course, proteins are made up of amino acids, but how do you think these amino acids are built? If you go back, what is that?
Uh, they're built by the DNA and RNA.
Wonderful. So that is what we call it as a DNA and RNA. So DNA is a genetic material. So it has got four bases, which we learned: A, T, G, and C. Okay, and these four bases, make up a single-stranded mRNA, where RNA is defined, with respect to the expressibility of the genome. Okay, which means the double-stranded DNA is broken down into a single-stranded RNA, which we call it as a messenger RNA, because an information is passed or message from DNA, which is double-stranded, to single-stranded RNA, and that single-stranded RNA gives rise to protein, which is made up of amino acids. And now comes a very important question: how do we know that this particular function of this particular protein is very important? How do we ascertain that particular functional entity of that particular protein that plays a very important role. Okay, so what happens is, imagine, a spring. Okay, imagine a spring, and imagine each part of that particular spring as a small, beads. Okay, or imagine, small pearl beads, which are kept on your table, and imagine each of those particular pearl beads, okay, as one amino acid. Okay, one amino acid. So let's imagine one amino acid, each are kept on the table. Now you don't know what to do with that, but as an intelligent person, what you do is you insert a small thread within that particular, protein molecules or amino acid molecules. Okay, and then you realize that this group of amino acids which make up the protein is now structured, isn't it? So now you realize that yes, a group of amino acids are intertwined together, and this intertwining gives rise to something what we call it as, a function. Yes, now maybe these particular beads can be used as, say, as a necklace or as a bracelet, okay, or something like that. So which means only when amino acids are structured together, you can ascertain the function of those particular, proteins. Okay, so that is where, the central dogma of bioinformatics comes up, and this is what we call it as holy grailing. Holy grailing is giving some kind of, information, giving some kind of, knowledge. So as you could see, you have got all the proteins which are made up of amino acids, which are put up like this, and if they are ordered, then only you can see a structure like this, which means, they can be folded. Okay, and once they are folded, the drug molecules can bind to that. These particular proteins that are present in our body. Okay, that particular drug can bind into these particular tiny pockets, as you can see. Okay, these particular pockets. Just because, this is bound, just because these are closely knitted together, then only a drug can come and bind this particular region. And then only you will know what is that particular drug acting upon that particular protein molecule, or what is the function of that particular protein molecule. Okay, so sequence. So once you have a sequence like this, a protein sequence, okay, you can predict this structure for this particular sequence. That's because you're putting it in an order, and once they're in an order, a protein can really fold because it is humongous, right? And now, because it is folding, you are giving some kind of a function. You're attributing some kind of a function to this particular protein molecule. Sometimes proteins में बहुत जिद्दी है, okay? वो बोलते हैं कि मैं fold नहीं करूँगा, ठीक है? मैं, if they are not folded, obviously, you don't get these pockets, right? मैं ऐसे ही रहूँगा. And that is where, if the proteins are not really folded properly, maybe that particular protein responsible for that particular organ or for that particular function, okay, is lost. तो यदि वो काम नहीं कर रहा है, तो then obviously, protein का function नहीं रहेगा, है कि नहीं? So that is the reason, why proteins, if they are folding properly, then you can definitely some kind of a function to that particular thing. उसको हम ordered protein कहलाएंगे, ठीक है? But हालांकि आपके पास ऐसे किस्म के proteins होंगे आपके body में, okay? Because हमारे body में 21.8 trillion cells होंगे, 21.8 trillion cells, okay? मतलब उतने trillion cells में उतने सारे proteins पैदा होगा, okay? उतना निक्षिप्त रूप से those particular proteins they are all sitting. Then each of those particular proteins should fold, but not necessarily all proteins would really fold. They might misfold or they might be disordered. Okay, but obviously when a protein folds, then only you can describe a function. And ऐसे protein information या DNA information अगर देखा जाए तो बहुत databases में it is stored. Then who checks these particular data databases? Obviously a bioinformaticist. Obviously a bioinformatician, okay, and that is why we people are here, okay. इतना humongous data कैसे check करना है? सारा चीज big data biologist check नहीं कर सकते, because big data specialist को biology जानते नहीं है, ठीक है? तो हम ऐसे लोग हैं जो biology थोड़ा बहुत जानते हैं, और ऐसे डेटा को कैसे इंटरप्रेट करना है, कैसे निकालना है, कैसे फंक्शनलाइज करना है, किस तरीके से फंक्शन को हम एक्टिवेट करना है, वो चीज हम कर सकते हैं, ठीक है। अगर आप देखेंगे तो ये सिंपलेस्ट चार्ट है। इसमें देखिए अगर आप 2023 की बात हो रहा है अभी, ये 2020 तक इतना डेटा, इतना बेसिस, डीएनए बेसिस, आरएनए बेसिस और सीक्वेंसेस आर नथिंग बट ए ग्रुप ऑफ बेसिस आर नथिंग बट एस टी सीस एंड जीस। वो इधर डीएनए के रूप में होंगे, आरएनए के रूप में होंगे या प्रोटीन के रूप में होंगे। तो आज देखिए मैं आज ही मैं ये वाला शायद मैं लिया था करीबन 9/16 को। तो इतने सारे मतलब 10 बिलियन सीक्वेंसेस है पूरा डेटाबेस में। मतलब 10 बिलियन सीक्वेंसेस को हम कैसे करेंगे, कैसे एनालाइज करेंगे, है ना। तो डेटा चाहिए और वी आर ऑल वर्किंग टुवर्ड्स समथिंग व्हाट वी कॉल्ड एज फाइटिंग टुवर्ड्स मूर्स लॉ। ओके, मूर्स लॉ एक चीज है, मतलब आप सब आजकल मोबाइल फोन्स और कंप्यूटर्स के जनरेशन वाले हो, यू नो हाउ टू यूज इट। पहले ऐसा चीज था, पहले 1 MB रैम कंप्यूटर्स होते थे। ओके, आजकल तो मिनिमम 16 GB रैम हम यूज कर रहे हैं। 1 MB कहाँ और 16 GB रैम कहाँ, है ना। तो आजकल मान के चलिए अगर आप 8 GB रैम अगर मिनिमम अगर आपका कंप्यूटर अगर यदि आप यूज कर रहे हो 2 साल के दौरान, मतलब से बाय 2025, ओके, जो 8 GB रैम से जो खरीदा हुआ कंप्यूटर, जो प्राइस जो था, सेम प्राइस 2 years आफ्टर भी सेम प्राइस रहेगा और डेटा डबल भी होगा। सो दैट इज व्हाट गॉर्डन मूर करके एक बहुत मशहूर साइंटिस्ट है। वो पिछले 4 महीने पहले उनका निधन हुआ, ये पास्ट अवे। सो ही पुट दिस पर्टिकुलर रूल, मतलब डेटा स्पीड के हिसाब से चलता है, डेटा स्टोर करने के लिए स्पीड के हिसाब से चलता है। तो उसी स्पीड से ये जितना भी जो डेटा है ये बहुत बहुत जीनोमिक डेटा जो है डीएनए के, आरएनए के, प्रोटीन के बहुत पैदा हो रहा है। देन यू माइट आस्क मी क्वेश्चन, ठीक है, डेटा पैदा कैसे होंगे, हाउ डू वी गेट दिस पर्टिकुलर डेटा। ओके, मतलब क्या चीज है जो डेटा, what makes that particular data in that thing special। okay, एक सिंपल चीज मैं आपको बताऊंगा। मान के चलिए देयर आर समथिंग कॉल्ड नोन नोन्स। नोन नोन्स का मतलब मान के चलिए एक प्रोटीन है या जीन है, अगर उसका फंक्शन यदि हम आपको पता चले तो उसका हम नोन नोन्स कहलाएंगे। ओके, likewise if there is a gene that is known and the function that is unknown, it is what we call it as known unknowns। okay, मतलब नोन नोन्स आप ये मान के चल सकते हो कि आपके जिगरी दोस्त है, ठीक है। ऐसे नोन अननोन्स होंगे कि आप सिर्फ बाय नेम आप जानते होंगे कि इनका नाम क्या है, बट यू डोंट नो व्हाट दैट पर्टिकुलर पर्सन डज इज और व्हाट दैट पर्टिकुलर पर्सन्स कैरेक्टर इज, और उनका हॉबीज क्या है आप जानते नहीं होंगे, ठीक है। ऐसे आप मान के चलिए। तो हमारे प्रोटीन्स या जीन्स जो भी हमारे बॉडी में होंगे, ये दो तरीके के होंगे, एक नोन नोन्स और एक नोन अननोन्स। ओके, और ये पूरा डिफरेंट कॉन्टेक्स्ट से बना हुआ रहता है और ये हम ओम्स कहलाएंगे, ओम्स या ओमिक्स। ओम का मतलब मेनी, ओके, ओमिक्स का मतलब मेनी या ओएमई का मतलब मेनी। जैसे जीनोम कहते हैं स्टडी ऑफ मेनी नंबर ऑफ जीन्स, प्रोटियोम्स स्टडी ऑफ मेनी नंबर ऑफ प्रोटीन्स। ओके, ऐसे होगा। ये चीज मैं आपको बाद में बताऊंगा। एंड नाउ कम्स ए वेरी इंपॉर्टेंट थिंग। अगर आप प्लांट की बात अगर आप करेंगे, can you tell me what is the difference between plants and animals? Anyone? Anyone? Plants और animals में क्या अंतर है, बता पाएंगे?
They both belong to different kingdoms, and there
Okay,
are a lot
yeah.
of differences on a cellular level amongst the two.
Okay, okay, very good. And what do you see physically? अगर physically अगर देखा जाए तो plants
are usually stationary while
very good.
animals do locomotion.
Plants are stationary, they are sessile. Okay, they cannot move. मतलब मान के चलिए ये मैं हूँ, I'm a plant, I'm just standing like this. I cannot move. अगर मान के चलिए अगर कोई वाटर अगर क्लॉग होता है, ओके, मेरे रूट्स में, प्लांट में, I cannot probably move, right? I cannot move like this, right? I have to do this, but उसका मतलब मैं स्ट्रेस में स्ट्रेस को मुझे कैसे ओवरकम करना है, मुझे सीखना पड़ेगा, है कि नहीं। तो कैसे सीखेंगे, वो स्ट्रेस के लिए एक जीन होगा, ओके, एक जीन या प्रोटीन होगा। दैट पर्टिकुलर प्रोटीन कोडिंग जीन या प्रोटीन या जीन वो प्लांट्स सीखता है, ठीक है भाई, देखिए अभी बारिश हो रहा है या पानी बहुत क्लॉगिंग हो रहा है, टेम्परेचर बहुत ज्यादा है, ह्यूमिडिटी बहुत ज्यादा है, ओके, और हमारे मेरे सॉइल के, मेरे रूट्स के बगल में बहुत सॉल्ट्स है, तो मैं सॉल्ट स्ट्रेस में कैसे झेल लूं, ओके। बट प्लांट्स आर सो इंटेलिजेंट कि दैट दे हैव देयर ओन मैकेनिज्म्स, उनके बॉडी में जो भी जीन्स जो होंगे वो निकाल देता है कि ये जीन, ठीक है, मैं ये काम में करूंगा, ये मुझे मेरा अभी मेरे रूट्स के जगह बहुत सारा सॉल्ट्स है, ओके, उसको मैं फाइट करूंगा, ओके। तो ऐसे तरीके के डिफरेंट ट्रेट्स बनते जाएंगे प्लांट्स में। ऐसे ही एनिमल्स की बात करेंगे तो हम काऊ की बात करें या बफेलो की बात करें, ओके, वो मिल्क ज्यादा देता है। ऐसे के तरीके अगर ज्यादा अगर उनको अगर हम ग्रास देंगे तो ज्यादा मिल्क देने का चांस है, ओके। ऐसे ही मिंक मिंक करके बहुत बहुत ही बढ़िया एनिमल है। किसी ने मिंक के बारे में सुना है क्या? एनीवन हैव हर्ड ऑफ मिंक? MINK?
Yes, sir.
Oh, very good. Yeah, mink is a wonderful animal. Its fur, its animal is made up of fur, which is used as a wool actually. Okay, जो हम sheep की wool ही use नहीं करते, डेनमार्क में जहाँ मैं था, और in Europe, they also use the fur, of that particular male. So they also have their own traits. और बैक्टीरिया की बात करें तो बहुत सारे बैक्टीरिया, जैसे अपर्णा आपको बताई थी, है ना, most of these particular bacteria are uh, uh, you know pathogenic, which means वो हानिकार है ना, और कुछ ऐसे बैक्टीरिया कुछ नॉन पैथोजेनिक होंगे, और कुछ मोनो आइसोलेट होंगे, मतलब एक ही आइसोलेटेड रीजन में रहेंगे, एक ही कॉलोनीज बनते रहेंगे, है ना, और कुछ होस्ट स्पेसिफिक है, मतलब कुछ ऐसे बैक्टीरिया सिर्फ ह्यूमंस में ही रहेंगे और कुछ ऐसे बैक्टीरिया सिर्फ काउस में ही रहेंगे, है ना। और ऐसे ह्यूमंस ह्यूमंस की बात अगर करें तो हम बहुत कॉम्प्लेक्स बंदे हैं। वी आर डैम इंटेलिजेंट, वी आर वेरी कॉम्प्लेक्स, वी आर नॉट ओनली मूवेबल, but we have learned how to move across in different conditions, and we are the only people on the earth probably where we can survive in -80 and also probably in +60 degrees Celsius as well, okay. Can you believe? Yes, that's surprising, isn't it? So that's because we have environment को हम झेल सकते हैं, that kind of complex traits हम बना चुके हैं, और वो चीज हम फिनोटाइप्स के रूप में, फिनोटाइप्स का मतलब जो बाहर फिजिकली आप कैसे हो, दैट इज व्हाट वी कॉल इट एज अ फिनोटाइप, ओके। फिजिकली कैसे आप दिखते हो, फिजिकली आप कैसे ये पर्टिकुलर चीज आप देख पा रहे हो, और कोई बंदा कोई मोटा है, ओके, उसका मोटापा कहते हैं, दैट्स व्हाट वी कॉल इट एज ओबेसिटी, ओके, एंड दैट इज अ टिपिकल फिनोटाइप। तो हर चीज अगर ये पर्टिकुलर थिंग्स में आप ट्रू पॉजिटिव्स देके जाएंगे, फॉल्स पॉजिटिव्स क्या है, ट्रू नेगेटिव्स क्या है, और फॉल्स नेगेटिव्स क्या है, ओके, ये शायद आपको ऐसे मिलेंगे, ओके। मतलब आप एक सुपरमार्केट जाइए, कम से कम एक प्रोडक्ट के लिए 4 वेराइटीज आपको मिलेंगे, ओके, और एक एक वेराइटी, एक एक डिफरेंट टाइप ऑफ मटेरियल होगा, डिफरेंट कॉस्ट होगा, ओके, और डिफरेंट व्हाट यू कॉल फंक्शनैलिटी होने का चांस है। तो आप आइंदा नेक्स्ट टाइम आप यूज करने के दौरान ही आपको पता चलेगा कि ये पर्टिकुलर प्रोडक्ट अच्छा है कि नहीं, ओके, दैट इज हाउ इट इज। सो व्हाट आई एम जस्ट ट्राइंग टू टेल यू इज बायोइन्फॉर्मेटिक्स is a tool where we study about this complex phenotypes, different phenotypes and traits के बारे में हम सुनते हैं। बट ये सुनने के लिए या देखने के लिए या करने के लिए, ओके, और दिखलाने के लिए, और वॉट एवर यू आर ट्राइंग टू डू, ये अनोखा चीज है। मतलब आप पिछले अगर आप 100 साल अगर आप देखेंगे तो सिस्टम्स बायोलॉजी तो बहुत जारी है, इट्स नॉट ए न्यू डिसिप्लिन, ओके, इट्स जस्ट लाइक एन ओल्ड वाइन इन ए न्यू बॉटल, okay, it's just like honey which doesn't get spoiled at all, okay. That's because बहुत सारे mutations, मतलब आज अगर आप कोविड की बात करेंगे तो करीबन 17 वेरिएंट टाइप्स हैं, मतलब 17 टाइम्स that particular छोटा 29,900 basis of DNA has been mutated, और 17 किस्म के वेरिएंट्स अभी मिल चुके हैं, अल्फा करके, ओमाइक्रॉन करके, डिफरेंट थिंग्स, मतलब डिफरेंट स्ट्रेन्स हैं, डिफरेंट वेरिएंट्स हैं, और बॉडी में बहुत सारे मॉलिक्यूल्स लाइक DNA, RNA और प्रोटीन, दे कीप ऑन म्यूटेटिंग, ओके। proteins, they keep on evolving. Okay, और कभी कभी अगर देखा जाए तो सेम जीन हानिकारक नहीं होने का चांस है, जैसे 20 साल पहले जब हुआ। अगर आप कोविड की बात करेंगे तो आज सार्स कोविड क्यों क्यों कहला रहे हैं उनको, क्योंकि 2004 में करीबन 20 साल पहले की बात था, सार्स कोविड 1 करके एक था उनकी जो रिलेटिव। ओके, सार्स कोविड 2 की अभी जो कोविड हम कहला रहे हैं उसके जो रिलेटिव। ओके, ड्यूरिंग दोज डेज इट्स द सेम सीक्वेंस थोड़ा म्यूटेट होके थोड़ा स्ट्रॉन्ग बन के अभी बहुत लोग को पिछले 3 साल में बहुत नुकसान किया। ठीक है, सो ये सारा चीज हम सिस्टम्स बायोलॉजी में हम सीखते हैं। ठीक? और ये चीज अगर आप देखेंगे तो सिस्टम्स बायोलॉजी ऐसी चीज है जो कैसे बॉडी में जितने भी जो बॉडी पार्ट्स है या डीएनए आरएनए और प्रोटीन है इंट्रैक्ट कैसे करेंगे। जैसे कार में ऐसे बॉडी पार्ट्स होंगे, डिफरेंट बॉडी पार्ट्स आर क्लोजली निटेड टुगेदर। अगर एक बॉडी पार्ट भी अगर हम निकालेंगे तो वो कार काम नहीं करता है ना। तो ऐसे ही ऑर्गेनिज्म में भी डिफरेंट ऑर्गन्स होंगे, ऑर्गन्स में ऑर्गेनेल्स होंगे, जैसे माइटोकॉन्ड्रिया हो गया है ना, क्लोरोप्लास्ट हो गया प्लांट्स में है ना। तो ये सारा चीज हम बायो इन्फॉर्मेटिक्स जनरेटेड सिस्टम्स बायोलॉजी में हम सीखेंगे। तो मैं आपको जैसे बताया था, ओमिक्स मतलब मेनी, जेनोमिक्स मतलब जेन इज स्टडी ऑफ मेनी नंबर ऑफ जीन्स, proteomics is study of many number of proteins, metabolomics और metabolomics दो चीज है। metabolism is constructive process और destructive process। constructive process is anabolome। okay, मतलब जैसे मान के चलिए मैं हूँ, अगर मैं ब्रीथ कर रहा हूँ, so I'm trying to construct some energies, I'm constructing some energies, that is anabolism। okay, and when I exhale, that is I'm destructing, which means whatever energies that are inside, I'm trying to dissipate it. Okay, of course, whatever I'm trying to breathe out carbon dioxide, but that is dissipated, that is a destructive process। इन दोनों को एनाबोलिज्म और कैटाबोलिज्म कुल मिला के मेटाबोलोमिक्स कहते हैं, है ना। एंड देन ये पूरा इंट्रैक्टोमिक्स मतलब हमेशा ये जितने भी जो मॉलिक्यूल्स होंगे, प्रोटीन्स है या डीएनए आरएनए है, ओके, 3 डिफरेंट टाइप्स ऑफ फ्रेंड्स मान के चलिए, ये हमेशा दे कीप ऑन इंटरेक्टिंग, देन ओनली इंटरेक्ट होने से ही दैट पर्टिकुलर फंक्शन होने का चांस है। ओके, अगर इंटरेक्ट नहीं होगा तो देन देयर इज नो फंक्शन, दैट इज डिसिपेटेड। ओके, फंक्शन आने का चांस है। ओके, सो ये क्या है, मैं आपको एग्जांपल के रूप में मैं आपको बताऊंगा। एनी क्वेश्चंस until here, dear friends? Yes. Any questions? Yeah, welcome, and Kiran. Yeah, sorry, Nitya. Yeah.
I'm sorry, I actually got slightly disconnected and was not able to hear the last thing you said properly.
Okay, yeah, I'll repeat it. Don't worry. Huh? Yeah, even the last slide, right, Nitya?
Yes.
Yeah, I'll do that. Yeah. By the way, your profile photograph is very good. Yeah.
Thank you, sir.
Okay. Yes. So where were we? We were here somewhere. Yes. So what I was just trying to tell you is omics is nothing but many. Okay. So whenever you say genomics, which is nothing but study of many number of genes. Okay. Proteomics is study of many number of proteins. Metabolome is nothing but anabolism and catabolism. Anabolism is always constructive process. Okay. If a plant is taking up certain water inside its xylem and phloem or inside its nervous system, okay, it is nothing but an anabolism. It is trying to construct some processes so that the plant can sustain. Likewise, I'm breathing some fresh air, that is what I'm trying to bring some constructive processes, that is what we call it as an anabolic process. Okay, likewise, when I'm breathing out whatever bad air is there, I'm trying to give it out, that is what we call it as a destructive processes. Okay, so interactions takes place. Functomics is nothing but study of many number of functions. Matlab, only when you have these molecules, they keep on interacting with each other. Not necessarily protein के साथ ही interact हो, या DNA के साथ ही interact हो. Protein can interact with the DNA can interact with RNA can interact with protein. Okay, then only a functional entity is maintained. मतलब तभी एक organism में कोई भी function अगर करना हो तो that particular function is retained. Okay? So यहाँ देखिए, यहाँ कुछ known अभी हम we learnt about known knowns and known unknowns, right? I told you known knowns का मतलब known is nothing but आपका friend है, नाम जानते हैं और उनका hobbies भी आप जानते हैं, है ना? You know what is that particular person, who is that particular person, you also know the hobbies. Known unknowns are those ones where you know that particular person's name, but you don't know what that particular person does. Okay, you don't know his hobbies, you don't know his demeanor, you don't know his anything. Okay, so those are known unknowns, और उनको हम hypothetical कहलाएंगे. Okay, so organism में, you know, if you have some known gene like this, देखिए कुछ gene का नाम है, इसका नाम है mutasB करके कोई है, या protein का नाम है. Okay. If it is interacting with several proteins within the cell, as I told you, there are about 21.8 trillion cells. उतने trillion cells में same genetic material replicate होगा, है ना? And they keep on interacting. A protein could interact with another protein, this protein, that protein, this protein, and this protein. They keep on interacting with each other. Okay, and then only they have to give rise to a function. It's just like, you know, shaking hands with each other. Suppose you are in a classroom. Okay, it's a new class, and you want to see how your friends are, you know, talking to each other. How do you start? Okay, you say that, yes, oh, I'm Prash. Okay, pleasure meeting you, Kiran. Nice meeting you, Jaraman sir. Okay, so likewise, you know, you shake your hands. So then only you know each other. So in a body as well, in a cell as well, you have these particular proteins that keep on interacting with each other. So when this particular protein is known to be interacting with this particular protein, okay, so for example, since this particular protein do not have any function, this particular protein, because just it is interacting, just because it has shook hands with this particular protein, it can derive the knowledge of what this particular protein is doing. Okay, so this particular information, the function of this particular protein, you know, could be derived. You know, the function of this particular protein could be derived from this. Okay, could be transferred from that. Okay, so now then you might ask me a question. कि आपके, you know, proteins interact क्यों होना है? Why do you think, you know, that the proteins, you know, should really interact? What makes that protein interaction very important? Okay, I think, you know, this is a class 9 or class 8 thing. Have you ever heard of enzyme lock and key model? Anyone? Nitya might have heard.
No, sir, I haven't.
Okay. Anyways, so very simple. Okay, so you have certain, you know, a protein like this. मैं आपको बताया था, protein has got, when it folds, you've got a lot of pockets. Okay, those particular pockets. Now what happens is, you know, a drug molecule or any other molecule can bind and come like this. So what it does is, this particular thing can really fit like this. Okay, or maybe, you know, it can fit like this, or it can fit like this, or it can fit like this, or it can fit like this. So you have certain spaces, you have certain grooves, just like, you know, a lock. What you can see, you know, ताला में, you know, कैसे, you know, space एक generate होगा. This particular space में, you know, एक और molecule can find and can fit in that, so that function of that particular protein can be done properly. Okay, मतलब कोई भी अगर protein molecule है, if it has to do the function, that particular protein cannot do the function alone. It has to invite someone. Okay, it's just like a fat man sitting like this. Okay, he has to move, or he has to, you know, move from one end to the other end. He cannot get up all together. He needs some help. Okay, he needs maybe help of another person. Maybe a tiny man, you know, has to lift in a big, you know, thing. He cannot do anything. So he has to join hands with another person, and then only he can do that. So this model is what we call it as a lock and key model. Okay, and आप जानते होंगे all enzymes are proteins, right? तो ये शायद, you know, अगर आप basic 10th शायद, you know, हमें तो 10th में ही पढ़ाया गया ये वाला, I don't know. I think, you know, it's a very simple model. This is how the protein looks like, and all the enzymes, you know, they can have, you know, that this thing, and all these particular proteins, when they keep on interacting with each other. This is how a protein interaction looks like inside the cell. This might be a bit overwhelming for you, but मैं आपको very simple के रूप में मैं आपको बताऊंगा. Okay, जहां भी red वाला है, okay, जो भी red, irrespective of whether or not the box type, it is a square or a circle or whatever, okay, wherever there is a red color, that indicates that that particular protein is a deceased protein. Okay, and wherever the protein, wherever the box type is yellow in color, okay, there is a mitochondrial protein. You know what does mitochondria does? If you are breathing fresh air, this is thanks to mitochondria. There are at least 2 to 3,000 mitochondria per cell. Okay, healthy mitochondria. If you are not able to breathe properly, then कुछ लगता है, कुछ problem है आपके cells में, mitochondria, they are not properly doing that. Okay, and these are all say mitochondrial proteins. And now you see a question. So if there is a red one with yellow one, इसका मतलब this is a deceased protein, okay, which means it causes disease, but it is also a protein that is associated with mitochondria. So what we did was a very simple question. We wanted to see, ठीक है, body में इतने proteins होंगे, we'll ask them to interact. Okay, we have developed a kind of a model. कुछ bioinformatics का model हम develop कर सकते हैं. Okay, and when we do this, several proteins, they keep on interacting with each other. Okay, and now if you see, not necessarily, say for example, if this is my protein, okay, of my interest, if this is interacting with this protein, and this protein is interacting with this protein, matlab if it is you, if this is your friend, and your friend might be having another connection here, okay, and this friend might be having another connection. But if this is the main protein of your interest, or if this is the main protein of interest, this protein and this protein are not directly interacting with each other, but it is interacting via another protein. Okay, so you will know about this friend only through this particular intermediary protein. Okay, so even in cells as well, the protein functions are described like this. Okay, matlab first degree, second degree, matlab friends of friends, friends of friends of friends. Okay, and now if you zoom in, okay, there might be several, matlab this particular friend might also be having another interacting partner somewhere. Okay, in another cell. So if you zoom in, the interaction keeps on increasing like this. Okay, can you see this is the difference between this and this, and these are all different proteins that are present in the cell, which does different types of functions inside the body. Different types of function, अलग-अलग proteins अलग-अलग किस्म के functions वो करते जाएंगे. Okay, and they're very unique. They do their own jobs. How to do that. Then how do we know that those particular proteins are doing that kind of function? Okay, then there are tools. Bioinformatics में कुछ tools हैं, like gene mania करके, string db करके. As a part of our exercises, I'll tell you during the next 15 minutes. Okay, so that you can do these exercises. Okay, how do you determine that a protein same function करने का chance है? मैं आपको बताऊंगा. Okay, so for example, this is my office. Okay, you can see my office. This is my office. I'm only, I'm the only person as of now sitting here. Okay, and now let's imagine I want to talk to a person sitting in another office somewhere there. There's another office over there. Okay, I cannot talk like this because beach mein divar hai, beach mein something is there. Okay, I need to either go to that particular office and talk. Isn't it? मतलब I am localized in one, office or one organelle. मतलब ये इतना मेरे बड़े बिल्डिंग में मैं तीसरी मंजिल में हूँ, थर्ड फ्लोर में हूँ. ओके, थर्ड फ्लोर में एक क्लास में या एक ऑफिस में मैं बैठा हूँ. If I want to talk to another person, I need to go to this office. Likewise, हमारे बॉडी में बहुत सारे सेल्स होंगे. मैं आपको बताया था 21.8 trillion cells. Can you believe 21.8 trillion cells? And each of these trillion cells has got some proteins. And not necessarily all the proteins would be localized only, in a small room like this. Imagine I'm a mitochondria. This office is mitochondria and I'm a protein. Okay, and I will only talk with my protein sitting in here. Okay, if I were to talk to a protein outside mitochondria, I'll go outside, okay, and talk to that particular person. But again, my home is this only. I should again, come back. Okay, I cannot, stay outside my office. Okay, body में भी ऐसे ही होता है. और एक तरीके में अगर एक टाइप में अगर ये पर्टिकुलर प्रोटीन इफ ऑब्वियसली फॉर सम रीजन अगर उस सेल उस ऑर्गेनल के बाहर अगर यदि जाता है तो देन सेल में बहुत सारे फ्लक्चुएशंस होने का चांस है. ओके, सेल बिहेव्स वेरी डिफरेंटली. अगर सेल बिहेव्स डिफरेंटली देन वो टिश्यू मतलब वो किडनी या लंग्स या हार्ट, ओके, इट बिहेव्स डिफरेंटली. और यदि वो डिफरेंटली बिहेव करता है तो ऑब्वियसली द एंटायर ऑर्गेनिज्म, एंटायर ह्यूमन बीइंग might behave differently. Okay, and as a result the person dies. Okay, so why should a protein interact? Why should a particular protein interact with a specific other protein only? Okay, ये चीज हम सीखे हैं because of known knowns and known unknowns. So हालांकि बायोइन्फॉर्मेटिक्स ऐसी चीज है where you can do experiments और बायोइन्फॉर्मा ट्रिक कर सकते हैं. ओके, मतलब you need not do all those experiments, but आप बायोइन्फॉर्मेटिक्स से क्या है, अगर आप बड़े होने के बाद if you want to become a bioinformaticist, okay, you can make your task very easy. Okay, sitting before your computer you can design your experiment, you can plan your experiment, okay, and then you can come up with a wonderful hypothesis. Okay, ये हम यूज करेंगे, but I'll not probably go in detail. I'll probably stop here for theory unless otherwise you have some questions. Any questions, please? Yeah.
Sir, like you mentioned, that enzymes are essentially made out of proteins. So, in the stomach, we have an enzyme called pepsin, which acts on proteins. So, how is it that it's a protein that is helping digest another protein?
Yeah, that's because there is what if in a protein, since a protein is made up of amino acids, which is an enzyme, isn't it? Now, in order to have that particular enzyme, derive some function, it needs the help of another enzyme, which is again a kind of a protein. Okay, that particular protein has to sustain. It has to generate, something, isn't it? It has to do something exciting. Okay, and in order to do that something exciting, it takes the help of another, close relative of that particular pepsin. Okay, and both of them, they repair that particular that whatever function, or they do that particular function. Then how do you do that particular function? Because it cannot say that ठीक है भाई तुम आ जाओ करके ऐसे नहीं बोलता है. Okay, there is always a space, and what is that space? ठीक है, I'll come and sit like this, you come like this, and I'll probably we'll go together like this, and we'll perform this particular experiment like this, or we'll do that particular repair, that particular thing like that. Okay, and now comes a very important thing. ये पूरा function, ये particular pepsin or that particular digestion पूरा होगा. Okay, and then ठीक है, then it says that ठीक है मैं अलविदा कह रहा हूँ, my job is done. Okay, and now can I leave? ठीक है भाई, I'm now leaving, and it leaves. Okay, so whenever कोई भी अगर repair होने का chance है, जहाँ भी repair होने का आवश्यकता है, okay, all these particular proteins are wonderful friends. Okay, even with a single whisper, they come together. Okay, and they recognize, they know that certain proteins need certain help. Okay, that kind of imaginative thoughts, these particular wonderful proteins, they have it. Okay, is that clear, Nitya?
Yes, sir.
Yeah. Any questions from Usma's daughter? Yeah. कि बेटा आपको समझ में आया? Okay, yeah, I'll just probably go ahead. So now let me open something what we call it as Gene Mania, which is a very simple tool. Okay, Gene Mania मतलब gene को हम इंट्रैक्ट हो रहा है कि नहीं, इसका फंक्शन क्या है, हम प्रेडिक्ट कर सकते हैं. ठीक है. Now you see, this is organism. These are different organisms, right? Arabidopsis thaliana, which is a well-known plant. It's a cress plant. Yeah. So what is all model organisms, in fact. Okay. C. elegans. Okay. C. elegans is a well-known worm. Okay. Danio radio. Okay, which is a fish. Drosophila melanogaster, which is a fly, E. coli, Escherichia coli, which is bacteria, Homo sapiens is human beings, Mus musculus, okay, is the mouse, Rattus norvegicus, which is nothing but lab rat, okay, and Saccharomyces cerevisiae, which is a budding yeast. Okay, these are all some 9 or 10 different organisms. और हर एक ऑर्गेनिज्म में हम प्लांट्स हम different proteins हम identify कर सकते हैं. Okay, so for example, since we are humans, let us search the human proteins, and I'm a bit selfish. I want to search some human proteins. Okay, can anyone tell me any of your favorite proteins of your interest? Anyone has got any protein to which you want to identify, whether or not a protein is interacting or not? Yeah. Suppose I'm taking some insulin protein. Okay. Insulin is a protein called INS. Okay. When you type the full name, usually आता है, or otherwise you type this. Usually protein or gene names usually 3-letter के होंगे. Okay. So this is an insulin protein. Okay. And it says that source is HGNC. HGNC is Human Genome Nomenclature Classification System. मतलब gene name कैसे देंगे? It will the consortium will tell you. Okay. Anyways, don't get into detail. Huh? The HGNC is this insulin करके एक प्रोटीन का नाम दिया हूँ, और सिंपली मैं सर्च करना चाहता हूँ कि ये प्रोटीन आखिर क्या है, कैसे what are the interacting proteins, I'll be able to know. Okay, let me see this. Now you see, these are all the interacting partners. Imagine you are a cell. In a cell, in a protein cell, you have got all these particular things. Okay, now you click this, the circular one. Okay, so if you click this, ये पूरा arrange हो जाएंगे ऐसे. And this is your protein, which is an insulin protein, which is at the center. Always your query proteins are usually in very big color. Okay, or color of these particular proteins, उनको हम nodes कहलाते हैं, nodes or protein names, whatever. Okay, because they're all interacting. Okay, और जो lines है बीच में, इसको we call them as an edges. Okay, so this is a node. These are all nodes, and the lines are called as edges. Okay. And there's just like these lines are connected together through with the nodes, or nodes are connected together with these lines. Okay, and now comes a question. Simple name. This is a pink one. Suppose you want to see only the physical interactions. Physical interaction मतलब दो proteins physically ऐसे interact हो रहा है. Okay, that is what we call it as physical interactions. Okay, so likewise co-expression. Co-expression is nothing but they are talking with each other at a given point of time, at the same time. Expression is nothing but talking, isn't it? So they're talking to each other. Likewise, co-localization. Co-localization मतलब sitting in the same organelle, which is मतलब दो proteins अगर co-localized है, इसका मतलब they're sitting in the same organelle like mitochondria or nucleus or cytoplasm or whatever. Okay. And fourth one is genetic interactions. They might be interacting with each other. Maybe they might be genetic. Genetic matlab they might be carrying the same disease. Okay, and same pathways. Same pathways का मतलब they might be doing the same functional modules. मतलब अगर कोई एक प्रोटीन है, एक प्रोटीन इंसुलिन पैदा होने के लिए अगर if it is doing, then all other proteins interacting with that particular thing will also tell the same. It also does the same. Okay, so shared protein domains मतलब एक प्रोटीन में usually, if you see this particular protein like this, okay, a protein structure में usually 3 domains होते हैं. 3 domains मतलब 3 parts. एक N-terminal domain like this. N-terminal मतलब जो यहाँ इस तरफ, okay. C-terminal मतलब usually 2 ends में जो first part को हम N-terminal कहलाएंगे और last part को हम C-terminal कहलाएंगे. और एक amino acid पूरा ये protein structure में होगा. But similar amino acids पूरा N-terminal में होने का chance है या C-terminal में होने का chance है. वो कैसे पता चलेगा? Based on protein domains. और कभी-कभी हम predict भी कर सकते हैं कि वो particular proteins are maybe similar and they might be predicting with each other. Okay. तो ये डिफरेंट टाइप्स ऑफ थिंग्स है। जैसे मैं आपको बता रहा था कि सारा प्रोटीन्स ही है, प्रोटीन्स मान के चलिए एक सेल में है, okay, and they keep on interacting with each other, they do some function, okay, and that function is very important. And how do you think that that function is done? फंक्शन तभी करेगा जब प्रोटीन्स फिजिकली इंटरेक्ट होने का चांस है, मतलब दे आर शेकिंग देयर हैंड्स। जैसे मैं आपको उस दिन बताया था, उस टाइम पे मैं बताया था कि एक प्रोटीन बताएगा ठीक ठीक है भाई, आपका सख्त जरूरत है, आप मुझे हेल्प करो, तो ये बंदा आएगा, इट विल फिट लाइक दिस, डज दैट पर्टिकुलर थिंग, ओके, एंड देन गोज बैक, ओके। इसका मतलब ये फिजिकली बॉन्ड है। और लेकिन कुछ प्रोटीन्स होंगे सिर्फ हेलो हेलो बोलेंगे, ठीक है भाई, ओके, हेलो, I need your help, okay, then I'll send you some other friend, okay, then that particular friend will come. That is what we call it as a co-expression, okay, मतलब ठीक है, मैं कुछ एंजाइम्स में कुछ चीज में इंड्यूस करूंगा प्रोटीन सेल में ताकि आपको हेल्प हो सके, okay, it's just like breathing in some oxygen, ठीक है, so then that particular person and all they do the same work, okay. और co-localization, कभी-कभी मैं आपको बताया था कि अगर दूसरे रूम में अगर कोई बंदा है, आप बात नहीं कर सकते हो, तो व्हाई नॉट यू थिंक दैट उस बंदा को बुलाया जाए यहाँ पर और बात करे, ओके, एंड देन यू थिंक दैट यू सिट हियर एंड रिक्वेस्ट दैट पर्टिकुलर पर्सन कि कैन यू कम हियर, विल टॉक टुगेदर। लाइकवाइज प्रोटीन्स आल्सो से दैट ठीक है, आई नीड सम हेल्प, ये कुछ मशीनरी मुझे रिपेयर करना है, आप वुड यू माइंड कमिंग हियर? तो वो बंदा क्या करेगा, दूसरे ऑर्गेनेल से आपका ऑफिस में आएगा, या आपके ऑर्गेनेल में आएगा, आपके माइटोकॉन्ड्रिया में आएगा, आपके न्यूक्लियस में आएगा, and does the function and then goes back. Okay, is that clear, dear friends? Yes. Anything else? So this is how the proteins are interacting, and then you can save this image. Wonderful network. और एक एक सिर्फ आपको फिजिकल इंटरेक्शन से देखना हो तो फिजिकल इंटरेक्शन से देख लीजिए, ओके। देखिए ये फिजिकल इंटरेक्शंस पिंक एडजस्ट सी है, पिंक लाइन से इंडिकेट हो रहा है, ये लोनर नोड है, ये नहीं है, ऐसे ये भी लोनर नोड है, ये भी इंटरेक्ट नहीं हो रहा है, ये भी लोनर नोड है, ये भी लोनर नोड है, ओके, और ये भी, ओके। तो ये चाहो तो आप निकाल सकते हो, बट ये दूसरे टूल्स से हम निकालेंगे, बट इसमें ये प्रोटीन सिर्फ जहां जो इंटरेक्ट हो रहा है, ओके, ये देखिए ये सिर्फ डायरेक्ट इंटरेक्टिंग पार्टनर्स है, ओके, ये भी नहीं है और ये भी नहीं है इनफैक्ट, है ना, और ये भी नहीं है, ये भी इंटरेक्ट नहीं हो रहा है आपका जो प्रोटीन्स है, सिर्फ यही हो रहा है, और ये भी इंटरेक्ट नहीं, और इसका भी उसका इंपॉर्टेंट नहीं है ना, लेकिन ये वाला सारे इनके साथ इंटरेक्ट हो रहा है, तो इसका आप बोल सकते हो कि ये सारे फिजिकली बाउंड है, मतलब ये दोनों, ये सारे प्रोटीन्स शायद एक ही किस्म के फंक्शन करने का चांस है, मतलब ये इंसुलिन रिसेप्टर है, ओके, इंसुलिन को रिसीव करता है इसलिए रिसेप्टर है। देखिए इंसुलिन रिसेप्टर, इसको अगर क्लिक करेंगे तो इंसुलिन रिसेप्टर है, ओके, तो बॉडी में इंसुलिन को पैदा करने के लिए मदद करता है। ऐसे डिफरेंट टाइप्स ऑफ प्रोटीन्स है, इसको आप क्लिक करेंगे, जैसे ये, इसको अगर आप क्लिक करेंगे तो इसका एंडोप्लाज्मिक रेटिकुलम ऑक्सीडाइज करके एक फंक्शन है, आईडी इंसुलिन डिग्रेडिंग एंजाइम, इट्स एन इंसुलिन डिग्रेडिंग एंजाइम। लाइकवाइज ये वाला सम अदर नेम, ओके, डिफरेंट प्रोटीन नेम्स। लाइकवाइज अगर आप फिजिकल इंटरेक्शंस नहीं चाहिए होगा, सिर्फ आपको को-लोकलाइजेशन चाहिए होगा। को-लोकलाइजेशन अनफॉर्चुनेटली देखिए कोई नहीं है, मतलब there are no proteins that are probably co-localized, I think, okay, मतलब they are not sitting in the same organelle, okay, but हालांकि 3.63% बता रहा है, but I don't know why. Can you see any lines here, small lines कहीं पे? No, okay, usually यदि नहीं है तो it is not, okay, and then pathways, अगर आप pathways करेंगे तो same function करने का chance है। देखिए pathways usually physical interaction से ही होगा, okay, अगर two proteins अगर physically interacted हो तो you can do that। और shared protein domains मतलब मैं आपको बताया था कुछ अमीनो एसिड्स एंड टर्मिनल में ही होंगे, ओके, शेयर होने का चांस है, तो वो सारे प्रोटीन्स आप देख सकते हैं। और कुछ प्रिडिक्टेड भी होंगे, मतलब प्रिडिक्टेड मतलब अगर आप ये क्लिक करेंगे, हमेशा ये इंसुलिन और ALX3 इसके साथ प्रिडिक्ट होने का चांस है। जहाँ इंसुलिन है, ALX3 या ये वाला प्रोटीन या IGF2, ये पूरा एक ही ग्रुप जैसा बनने का चांस है। ये जीन मैंने करके एक टूल ऑटोमेटिकली वो प्रिडिक्ट कर रहा है, ठीक है. सो नाउ यू कैन डू ऑल ऑफ देम, टेक इट, ओके. देन यू कैन मेक दिस सर्कल आएगा. उसके बाद ये नेटवर्क इमेज आप डाउनलोड करके आप देख सकते हो. यहाँ पर देखिए मैं डाउनलोड किया, इमेज ऐसे आ गया, वंडरफुल इमेज वेयर आई कैन कीप इट इन द फॉर्म ऑफ अ प्रेजेंटेशन. और एक रिपोर्ट अगर यदि मैं जनरेट करना चाहूँ तो रिपोर्ट जनरेट कर सकता हूँ रिपोर्ट करके, ओके. और ये मैं रिपोर्ट अगर मैं सॉरी रिपोर्ट डाउनलोड हुआ क्या, नहीं हुआ रिपोर्ट, ओके. 1 minute है ना, it's it's coming, 1 minute. सॉरी थोड़ा ये हो गया, आई एम जस्ट रिफ्रेशिंग, आएगा. ये जावा होनी चाहिए आपका कंप्यूटर में, तभी ये होगा. सो नाउ वी हैव गॉटन इट, टेक इट, सेव रिपोर्ट. Yeah, I think it's still downloading. It will take some time. Usually पता नहीं, या मेरा कुछ प्रॉब्लम है यहाँ पर. या अब हो गया, अब बन गया. तो देखिए ये जीन मैंने रिपोर्ट बन गया. दिस इज हाउ द नेटवर्क इज. ये डिफरेंट रेफरेंसेस, डिफरेंट पेपर्स, मतलब जो साइंटिफिक पेपर्स जो छापे हैं उसका डिटेल्स आएगा, ओके. डिफरेंट मेथड्स से आएंगे, ओके. डिफरेंट जीन्स ये हैं और रैंक भी है, मतलब इसमें ऐसी कौन सी रैंक है जो प्रोटीन टॉप रैंक है. तो ऑब्वियसली हमारा रैंक इंसुलिन ही है, फर्स्ट रैंक है, मतलब ये नॉट अवेलेबल है, बट इंसुलिन रिसेप्टर फर्स्ट रैंक होगा. बिकॉज ये नॉट अवेलेबल इसलिए और नॉट एप्लीकेबल इसलिए है बिकॉज ये हमारा क्वेरी प्रोटीन है, ओके. तो ये रैंक्ड प्रोटीन है, इंसुलिन रिसेप्टर फर्स्ट रैंक है, और बाकी अन्य प्रोटीन्स प्रोटीन के नाम होंगे. और डिफरेंट फिजिकल इंटरेक्शंस का रेफरेंस क्या है, मतलब फिजिकल इंटरेक्शंस अबाउट 77% ऑफ दिस नेटवर्क बन रहा है, ओके. ऐसे ही other interactions like एक मिनट नीचे अगर आप देखेंगे तो there could be other networks, बट देखिए यहाँ कोई एक्सप्रेशन है, कोई एक्सप्रेस के इतने सारे प्रतिशत के पेपर्स हैं, साइंटिफिक पेपर्स, जैसे कोलोकलाइजेशन, प्रिडिक्टेड, ओके. ऐसे डिफरेंट टाइप्स ऑफ डिटेल्स इससे हम जनरेट कर पाएंगे. इज दिस क्लियर डियर फ्रेंड्स? Yes, Nitya?
Yes, sir. I wanted to ask that in the PDF you just showed us, there was a percentage given for the physical interactions. Like, what does that exactly signify?
Yeah, that exactly shows that 77% of all these particular proteins that are interacting are physically associated. Okay, matlab there are 77% of these particular proteins. Okay, you see, out of so many proteins, only maybe this protein, or maybe this protein, or maybe this protein. Or
So
maybe
you're
this.
not presenting your screen.
Oh, sorry. Now, yeah, now it is visible, right? Yeah.
Yes, sir, it's visible.
Yeah. So here, when I click physical interactions, it indicates that 77% of all the proteins here are physically interacting with each other. And there could be this protein that is loner, that is not interacting. This is also a loner. This is also a loner. Okay. And there are very few proteins, that are not interacting at all. So only 77% of these particular proteins are physically interacting, which are shown in these, pink lines or pink edges. Okay, that is that particular percentage that is denoted. Likewise, 1.84%, matlab there could be only 1.84% of the proteins here, okay, that are responsible for making this particular proteins, perform some functions. Okay, Likewise, co-localization. There are about 3.63% of the proteins that are interacting or co-localized. Likewise, co-expression. Okay, about 8.01% of the proteins are co-expressed with each other. Okay. So that percentage indicates that that amount of percentage of proteins are known to be interacting with each other inside the cell. Okay.
Yes, sir.
Any questions here, please? Yeah. So I think if there are no questions, I'll probably call it a day today, assuming that you will go to genemania.org, okay, and do that particular exercises. Okay, take any protein of your interest. You can find any protein name across different organisms as well. Like for example, here I'll again share the screen. Here I've taken insulin, but you might also take in mouse as well. Mouse may be a particular protein होने का chance है. Okay. Okay, but mouse may it might be a different name. You need to be very careful. Okay, so you can take any other name like BRCA or BRCA1 or BRCA2, whatever. Okay, so this is a BRCA1, which is a breast cancer gene. Okay, in mouse, if you click and search this, you'll get some network. Okay, it will take some time because of Java, right? So when you can click this, it's a mouse. It's a mouse network. The same thing, you can also search in humans as well. Okay, humans में थोड़ा different network होने का chance है. Okay, that will also allow you to do that. So you can also do this. You see, in humans, the network. I think it's in humans. I clicked. Okay, you click search. You'll get this network. Okay, you have a bigger, denser network because humans may this particular protein के बारे में many have studied. Okay. You can do this, and you can see what are those particular protein names, and be inquisitive. If you go down the report, you can learn about that particular protein name. Okay. Sometimes that protein name may not be there, but so for example, if you know what is BARD1, you can also use something called genecards.org. Okay, and type that gene. Unfortunately, this is only for humans. Okay, BARD1 करके अगर आप सर्च करेंगे तो ये आपको बताएगा कि BARD1 क्या है. It's a BRCA1 associated ring domain. Okay, ये function क्या है आपको पता चलेगा थोड़ा बहुत. What does that particular function does. Okay, so I'll not get into detail. I'll probably stop here unless otherwise you think that you have that exercises done. I'll be very happy if you do go through that genemania.org and do those exercises. Okay. Any questions? I'll be very happy to answer. Otherwise, we'll call it a day.
No, sir, no questions.
Okay. So thank you so much. So with that note, I would like to thank Rajal for again, graciously spending her time in recording and managing these wonderful efforts. Thank you so much, Rajal.
Thank you, sir. Thank you for taking today's lecture.
Thank you. Yeah, thank you, Nitya and Usma's daughter. Thank you, Jeraman sir and Kiran. Thank you so much. Huh? Bye, all of you. Bye.
Thank you, sir.
Thank you, sir.